Usually, medical diagnoses feel a bit like engineering. Like if you break your arm, the X-ray shows this jagged white line across the bone, and the doctor just points at it and says, well, there's the problem, and here is how we set it.
Right. It's very visible, very binary.
Exactly. It's a visible, mechanical problem. But the moment you step into the world of molecular biology or, genetic diseases, that X-ray machine is completely useless. The landscape is entirely invisible.
Yeah, and because it's invisible, traditional pharmacology has essentially relied on, well, blunt force instruments for decades.
Right. We've spent, a century now just throwing chemicals at these incredibly complex biological systems. We're trying to plug a leaky pipe while trying very, very hard not to accidentally flood the entire house in the process.
And that flooding, that collateral damage of traditional medicine is something we've just kind of accepted as the cost of doing business, until very recently.
Because you're trying to stop a bad protein that's already out there.
Exactly. When you are trying to inhibit a fully formed, malfunctioning protein that is already wreaking havoc in the body, you inevitably disrupt other systems. We've been treating the symptoms of bad biology, not the root cause.
But today, we're doing a deep dive into source material that completely upends that entire paradigm. We are exploring how scientists took this really bizarre, accidental botanical discovery, something involving a white petunia, of all things.
Yeah, a flower.
Right, a flower. And they turned it into a programmable molecular robot, like a literal drug that doesn't just treat symptoms, but actively reads and deletes the fundamental code of human disease.
It's incredible.
We're diving into the mechanics of small interfering RNA, or siRNA. Okay, let's unpack this. We've got a huge stack of scientific literature, clinical trial data, historical accounts, and pharmacological analyses to go through.
And if you look closely at the overarching theme of all this research we're analyzing today, what we're really witnessing is the dawn of the information age of medicine.
The information age. What does that mean in this context?
Well, in this era, drugs are no longer just static chemical compounds that bump into things. They are active pieces of code.
Like software.
Exactly like software. They are engineered to intercept and destroy specific harmful biological messages before the disease even has a chance to physically manifest inside the cell.
So, to trace the origin of this technology, we actually have to step out of the high-tech pharmaceutical lab and go into a greenhouse, right? Like back in the late 1980s.
Yeah, it's a great story. Botanists were trying to execute what seemed like a very simple, logical genetic modification.
Right, they wanted to take red petunias and just make the petals even redder. And the logic they were operating under makes total sense, right?
Yeah.
Like, the flower produces a red pigment using a specific gene, the chalcone synthase gene.
That's the blueprint for the red color.
So they figured if one gene gives you a red flower, injecting extra copies of that exact same gene should just give you a brilliantly, intensely red flower, like more red paint in the bucket.
And that assumption perfectly reflected the central dogma of biology at the time. More genetic instruction should directly translate into more protein product.
Yeah.
You add more blueprints, the cellular factory builds more pigment.
But nature completely ignored the textbook. So they injected these extra copies of the gene into the petunias, and when the flowers finally bloomed, they weren't super red at all.
No, they bloomed pure white.
Pure white. Or sometimes they had these weird, chaotic, variegated, patchy patterns. It's as if adding 10 more blueprints to the construction site caused the builders to just panic and burn the entire site down.
That's a really good way to put it. How does that make sense? It was a paradox that completely baffled geneticists for years.
What were they calling it back then?
They initially gave it a descriptive name, co-suppression, because both the newly introduced gene and the plant's natural endogenous gene were being suppressed simultaneously.
Oh, so the plant was turning on its own natural red pigment too.
Exactly. And around the same time, researchers looking at fungi observed a nearly identical phenomenon, which they called quelling.
Quelling.
Yeah. The cellular machinery wasn't just ignoring the extra instructions, it was recognizing the sudden overabundance of this specific genetic code as a severe anomaly, like a threat.
So the cell essentially panics at the sight of too much identical code and just hits a biological mute button on the whole operation.
Right. It actively sought out and destroyed all messages related to that pigment.
But the actual mechanism, like what that mute button was physically made of, that stayed a mystery for another decade. right?
It did. It wasn't until 1998 that the pieces finally fell into place. And the breakthrough didn't come from plants or fungi. It came from Andrew Fire and Craig Mello working with a microscopic nematode worm.
Ah, Caenorhabditis elegans, C. elegans.
Yes, and their experimental design with C. elegans is really a masterclass in isolating biological variables. Fire and Mello wanted to know exactly what physical shape the genetic material needed to take to trigger this massive silencing effect.
Okay, so they focused on a specific target gene in the worms called PAR1, right?
Right. Now, to understand their experiment, we have to recall how genetic information flows. I mean, DNA is the permanent archive in the nucleus.
Like the master blueprint.
Exactly. When a cell needs to make a protein, it copies the DNA into a temporary single-stranded message called messenger RNA or mRNA. That mRNA is sent out into the cell to be translated into a protein.
Okay, so the mRNA is the ultimate target here.
Mm-hmm.
So Fire and Mello decided to inject different synthetic versions of this RNA into the worms to see what would happen.
Right. First, they injected the sense strand.
Which is just an exact replica of the target mRNA, right?
Yes. And the worms were totally fine. The gene kept working normally.
Okay, so then they injected the antisense strand, which is the mirror image sequence. And the thought process there was that the antisense strand might stick to the mRNA, like Velcro, and physically block it from being read.
Yeah, and that approach was actually a huge area of research at the time. A lot of people were betting on it. But Fire and Mello noticed it only had a very weak, inconsistent effect.
It didn't really work. The gene still mostly functioned normally.
Exactly. The true breakthrough happened almost by accident. You see, when you're synthesizing these single strands of RNA in the lab, it's very common to get tiny amounts of contamination.
Contamination? Like what?
Like where the sense and antisense strands accidentally bind together in the test tube to form double-stranded RNA.
Oh, so they purified that double-stranded RNA, injected just a tiny amount of that duplex into the worm, and what happened?
The result was catastrophic for the target gene. Absolute profound silencing.
Wow.
The double-stranded RNA didn't just block the target mRNA, it catalyzed its total disappearance from the cell.
So it wasn't just covering it up, it was actively destroying it.
Right, and the efficiency of it was staggering. Just a few molecules of double-stranded RNA were enough to completely shut down the gene's expression across the entire body of the worm.
Wait, really? Just a few molecules did all that?
Yes. And in some cases, the silencing effect was even passed down to the worm's offspring. This was the definitive discovery of RNA interference, or RNAi.
That is wild.
It was so completely unexpected and so monumentally important to our understanding of cellular biology that Fire and Mello were awarded the Nobel Prize in 2006.
Which is incredibly fast by Nobel standards. They usually wait decades.
Exactly. It was that big of a deal.
But, finding a mechanism like this sitting inside a petunia or a microscopic worm begs a massive evolutionary question. Why does this system even exist in nature?
It's a great question.
Because it's analogous to finding a highly sophisticated cellular spell checker that doesn't just highlight typos, but actively shreds the entire document. What is the cell guarding against?
It is guarding against some of the oldest, most insidious parasites on Earth. From an evolutionary standpoint, RNA interference is a primitive, highly conserved genetic immune system.
An immune system for genes.
Think about how certain viruses operate. Many dangerous pathogens carry their genetic code as RNA, not DNA. When they invade a cell and start rapidly copying themselves, they frequently form long stretches of double-stranded RNA.
As like an intermediate step in their replication cycle.
Yeah.
So double-stranded RNA floating freely in the cytoplasm is not normal biology.
Not at all. To the cell, it's a massive, flashing red alarm that screams, viral infection.
Okay, so that's the external threat. But what about internal threats?
The other major trigger comes from within our own genome. Our DNA is actually littered with things called transposons, or jumping genes.
Jumping genes.
Yeah, these are parasitic fragments of DNA that occasionally wake up, copy themselves, and insert themselves into new, random locations in our genome.
Which sounds like it could cause devastating mutations if they jump into the wrong spot.
It absolutely can. And transposons also frequently produce double-stranded RNA when they become active.
So over billions of years, these eukaryotic cells had to evolve a surveillance network to survive this. They built a system that constantly monitors the cytoplasm for double-stranded RNA.
Exactly. It reads the specific sequence of that alien RNA and then systematically hunts down and destroys any messenger RNA that matches it.
That is so cool. The virus is stopped before it can assemble, and the jumping gene is destroyed before it can jump.
The biological elegance of this defense system is what makes it so powerful. But, recognizing that double-stranded RNA is the trigger is only the first layer of the mystery.
Right, because discovering the phenomenon's one thing, but understanding the machinery behind it is another. If the double-stranded RNA you inject is just the signal, what is the actual weapon firing the bullet?
And that brings us to the actual cellular machinery of siRNA, the physical execution, how the cell actually chops up the invader.
Okay, let's break down this cellular assassin. The first step, the first protein that responds to this viral alarm, whether it's from a virus or our own jumping genes, is an enzyme called dicer.
Yes, dicer. Dicer is an RNase III endonuclease.
Let's define that. Endonuclease means what?
It means it cuts nucleic acids right in the middle of the chain, rather than just chewing them away from the ends.
Okay, so when dicer encounters a long molecule of double-stranded RNA, it doesn't just chop it up randomly like a blender.
No, not at all. Dicer physically acts as a molecular ruler.
A molecular ruler. Wait, how does an enzyme measure something? It doesn't have eyes.
It has to do with its physical three-dimensional shape. Dicer has a specific pocket called the PAZ domain that physically grabs the end of the double-stranded RNA.
Okay, so it anchors itself.
Right, and the actual cutting blades of the enzyme, the RNase III domains, are located a very specific fixed distance away from that gripping pocket.
Oh, so because of that fixed distance, dicer consistently chops the long double-stranded RNA into short, highly uniform fragments.
Exactly, fragments that are exactly 21 to 25 nucleotides long.
So no matter how long the invading viral RNA is, dicer runs along it and turns it into these precise 21-letter fragments.
Yeah.
And these fragments are the siRNAs, the small interfering RNAs.
Yes, and because of the specific way Dicer's blades slice through the double helix, these siRNAs are left with a very distinct physical signature.
What does that signature look like?
They have a double-stranded central region, phosphate groups naturally attached to their 5' ends, and a characteristic overhang of 2 nucleotides at their 3' ends.
So it leaves little loose ends dangling.
Exactly. And this isn't just a messy cut. That specific structure is a deliberate biochemical handshake.
A handshake?
It signals to the next set of proteins in the cell that this is a properly processed siRNA and it's ready to be loaded as ammunition.
Okay, so the ammunition is prepped. We've got these 21-letter siRNAs floating in the cytoplasm. Now the cell has to load them into the weapon.
Right.
This is the handoff to a massive multi-protein complex called RISC, the RNA-induced silencing complex.
And the core of that complex, the engine of RIC, the actual assassin holding the knife, is a protein called argonaute. In humans specifically, it's a GO2.
A GO2.
A GO2 is where the lethal intent of the system is realized. It possesses what we call slicer activity.
Slicer activity. I love how dramatic these biological terms are.
Biologists have fun sometimes, but before a GO2 can slice anything, it has to overcome a fundamental physics problem.
Wait, if the siRNA is double-stranded when dicer hands it over, how does it actually read the target mRNA? Doesn't it have to open up?
You identified the exact biophysical hurdle. The siRNA is a double-stranded molecule.
And so all of its binding sites are locked up, right? The base pairs, the A's, U's, C's, and G's are bound to each other. It's like a closed zipper.
Precisely. If it's closed, it can't read or bind to a new target mRNA floating in the cell. So the RISC complex has to physically pull the 2 strands of the siRNA apart.
It unzips it.
It unzips it. But it cannot keep both strands.
Why not?
Because if it kept both, it would be aiming at 2 totally different targets simultaneously, which would cause absolute biological chaos. So RIC has to select one strand to be the guide strand.
The antisense sequence that'll hunt the target.
Right. And the other strand, the passenger strand or sense strand, is just ejected from the complex and quickly degraded by the cell.
So it just throws away half of the material. But how does this non-conscious protein complex decide which strand is the guide and which is the trash? Is it just a random 50-50 coin toss?
Not at all. It is entirely deterministic, and the selection is based purely on thermodynamic stability.
Okay, let me push back on that term, thermodynamic stability. A protein doesn't have a tiny thermometer. How does a GO2 actually sense energy or stability?
It senses physical resistance. We have to look at the hydrogen bonds connecting the RNA letters.
Okay.
An adenine binds to a uracil using 2 hydrogen bonds, but a cytosine binds to a guanine using 3 hydrogen bonds.
Ah, so a sequence with more CG pairs is literally physically stronger and harder to pull apart than a sequence with AU pairs.
Exactly. So RISC essentially tugs on both ends of the tiny siRNA zipper.
That is a great visualization. It pulls on the ends.
And whichever end gives or unwinds first the end with the lower thermodynamic stability, meaning fewer hydrogen bonds, that dictates the orientation.
That makes so much sense.
The strand whose 5' end sits at that weaker, easier to unwind point is selectively retained by a GO2 as the guide strand. The asymmetry of the duplex ensures the weapon only points in one direction.
That is just incredible biological engineering.
Well,
So now we have a fully armed RASC complex. A GO2 is holding a single 21 nucleotide guide strand, exposing its sequence to the environment.
Right.
And it's just floating through the cytoplasm, acting like a cellular spell checker, scanning the thousands of messenger RNAs being pumped out by the nucleus.
And the scanning relies completely on Watson-Crick base pairing. The guide strand is looking for its perfect complement.
Like a key looking for a specific lock.
When it bumps into an mRNA sequence that is a 100% perfect match, the two strands snap together violently. They bind tightly across the entire 21 nucleotide length.
And that tight binding is the trigger for the assassin, right?
It is. The perfect geometric alignment of the guide strand and the target mRNA causes a conformational change.
A shapeshift.
A physical shapeshift in the AGO2 protein. This shift brings its catalytic slicer domain directly down onto the mRNA backbone.
Boom.
AGO2 cleaves the target mRNA exactly between the 10th and 11th nucleotides relative to where the guide strand is paired.
A perfectly precise molecular guillotine. And once that mRNA is sliced in half, it loses its protective caps.
Right.
Right?
Yeah.
The cell realizes it's just broken garbage and sends in enzymes to dissolve the rest of it.
Exactly. The mRNA never makes it to the ribosome, and the disease-causing protein is never built, gene silenced.
It's beautiful.
And what makes this highly efficient as a potential therapy is that the RISC complex is a true catalyst. It is not consumed in the reaction.
Wait, really? It can fire more than once?
Oh, absolutely. After it slices the target mRNA, AGO2 releases the broken fragments. The complex is immediately free to hunt down the next copy of that same mRNA.
Wow.
A single loaded RISC complex can destroy hundreds or even thousands of target messages.
So while we're looking at the mechanics of RASC, the source material draws a very sharp distinction here. It talks about siRNA, which is what we're discussing, and a related molecule called microRNA, or miRNA.
Yes, that's an important distinction.
If they both use this exact same RISC machinery, what actually makes them different?
Well, what's fascinating here is that the distinction dictates how we use them therapeutically. Both are small RNAs, and both load into Argonaut proteins. But the rules of engagement are entirely different.
Wow, so?
siRNA, as we've established, demands perfect sequence complementarity. It is a precise sniper. One exact match triggers the slicer activity and immediate target destruction.
But microRNA is less strict.
Much less strict. MicroRNAs are endogenous. Our cells naturally produce hundreds of different microRNAs to regulate our own biology.
Okay.
A microRNA only requires a partial match to its target. It primarily relies on just a tiny region at its 5' end called the seed region, which is usually just nucleotides 2 through 8.
So it only needs a 7-letter match to bind.
Right. And because it only needs a 7-letter match, a single microRNA might stick to dozens or hundreds of completely different messenger RNAs.
Oh, wow. So if siRNA is a sniper rifle hitting one specific gene, microRNA is like throwing a wide net over an entire biological pathway, like maybe just gently turning down the volume on 50 different genes at once.
Exactly. And the consequence of that partial match is that it usually doesn't trigger a GOCO2 to slice the mRNA. The geometry just isn't perfect enough to trigger the guillotine.
So what does it do if it doesn't slice it?
The RISC complex just sits there. It physically blocks the cell's machinery from reading the mRNA. We call that translational repression. And it sits there until the mRNA eventually decays on its own.
Okay, so microRNAs are broad network regulators, but siRNAs are engineered tools for absolute single-target precision.
Precisely.
Okay, so let's pause and appreciate the timeline here. Because by the early 2000s, the scientific community fully understood this mechanism. We had discovered this natural, devastatingly precise molecular guillotine sitting dormant inside every single human cell.
We had the blueprint.
Right, so we have a natural cellular sniper rifle. All we had to do is synthesize the ammunition, a 21-letter sequence of RNA matching a disease gene, and inject it into a patient. So why couldn't we just inject synthetic siRNA into a patient in 1999 and cure diseases immediately? Why did it take another 20 years?
Because the environment inside the human bloodstream is incredibly hostile to naked RNA. It is quite literally a biological war zone.
Well, We mentioned earlier that our immune system evolved to hunt down foreign RNA viruses. That must mean our blood is absolutely saturated with defenses.
It is. Our plasma and our tissues are swarming with aggressive enzymes called ribonucleases, or RNases.
And what do they do?
Their entire biological purpose is to find free-floating RNA and shred it into basic nucleotides. If you take unmodified raw synthetic siRNA and inject it into a patient's vein, it is like sending a paper boat into a hurricane.
It just gets obliterated.
It has an in vivo half-life of less than 5 minutes. It is completely annihilated before it ever reaches a target tissue.
5 minutes, that is useless as a drug. And even if you pushed a massive dose and a fraction of it survived the shredders, the immune system itself would completely overreact, right?
Oh, absolutely. The innate immune system utilizes pattern recognition receptors, specifically things called toll-like receptors, or TLRs, which are located on the surfaces of immune cells.
And they are actively scanning the blood for foreign nucleic acids.
Yes, injecting large amounts of synthetic, unprotected double-stranded RNA triggers these receptors. It initiates a massive cascade of inflammatory cytokines, a huge interferon response.
Which would make the patient incredibly sick.
It can cause severe shock-like toxicity. It's dangerous.
So you have a drug that disappears in 5 minutes, and in those 5 minutes it triggers a potentially lethal immune reaction. This bridges us directly into the need for chemical engineering.
The biologists had to step back and hand the problem over to the medicinal chemists. We had the logic, but we needed to build armor.
We needed to engineer a molecular robot that could survive the blood, hide from the immune system, and still somehow perfectly load into the RISC complex.
And this transition from biological discovery to chemical engineering is where the siRNA story becomes truly miraculous. The chemists were given 3 very clear mandates.
Okay, what were the engineering objectives?
First, stabilize the RNA backbone against those circulating RNA shredders. Second, cloak the molecule from the innate immune sensors, like the toll-like receptors. And third, sharpen the thermodynamic binding profile so the guide strand locks onto its target with extreme affinity.
Let's talk about this specific armor plating they designed, starting with the sugar ring of the RNA itself. Why focus on the ribose ring? What is the actual vulnerability there?
The fundamental Achilles heel of the RNA molecule is baked right into its name: ribonucleic acid. On the ribose sugar ring, attached to the 2' carbon position, is a hydroxyl group.
An oxygen bound to a hydrogen.
Right.
And why is that specific oxygen-hydrogen pair such a huge problem?
Because it is highly chemically reactive. In the presence of circulating RNase enzymes, that 2' hydroxyl acts like a molecular crowbar.
A crowbar?
Yeah, the enzyme utilizes that hydroxyl group to initiate a nucleophilic attack on the adjacent phosphate bond. It uses it for leverage to snap the RNA backbone in half.
So the most logical engineering solution is to just remove the crowbar so the enzyme has no leverage.
Exactly. The chemists began replacing that vulnerable 2' hydroxyl group with other chemical structures. And the 2 most vital substitutions they developed are 2-O-methyl, often abbreviated as 2-O-MA, and 2-fluoro, or 2F.
Let's break down the methyl modification first. What does adding a methoxy group do to the physics of the molecule?
By replacing the small hydroxyl group with a bulky methyl group, you accomplish 2 critical things. First, the sheer physical bulk of the methyl group sterically hinders the RNase enzymes.
They physically cannot get close enough to attack the backbone.
Right, the stability skyrockets. But second, that methyl group acts as a chemical disguise.
A disguise against those toll-like receptors we talked about.
Precisely. The toll-like receptors evolved to recognize native, unmodified viral RNA. The bulky 2-MO modification changes the surface topology of the molecule just enough that the immune receptors no longer recognize it as foreign.
Oh, wow.
The RNA effectively becomes invisible to the innate immune system.
That is incredible. It's armor and a cloaking device in one simple substitution. Okay, so what about the 2-fluoro modification? Why use fluorine?
Fluorine is a really fascinating element in medicinal chemistry because it is highly electronegative. When you replace the hydroxyl group with a fluorine atom, that electronegativity subtly shifts the entire physical conformation of the sugar ring.
It changes its shape.
It locks the ring into a shape that is thermodynamically primed for binding.
Going back to our earlier discussion about thermodynamics, it changes the energy required to pull the strands apart.
Yes. It significantly increases the melting temperature of the RNA duplex. The modified sequence binds to its target mRNA with far greater affinity. It locks on and refuses to let go, which dramatically amplifies the potency of the drug.
Now, my instinct as an engineer would be, I mean, if fluorine makes the drug that much more potent, why not just build the entire 21-letter sequence out of 2 fluoronucleotides? Make the most powerful sniper bullet possible.
It is a totally logical leap, and it's exactly what some early pharmaceutical companies attempted. But this brings us to a really harsh lesson in toxicology.
There's always a catch.
There is. While 2-fluoro vastly improves potency, pushing the system too far disrupts normal cellular biology. The sources specifically highlight an early-generation drug candidate called revusiran.
What happened with revusiran?
Revusiran relied very heavily on 2F modifications across the molecule to maintain its stability. It entered phase 3 clinical trials with very high hopes, but the trial had to be abruptly halted.
Halted? Why?
Due to unexpected clinical toxicity and, sadly, increased mortality in the treatment group.
Wait, was the fluorine itself poisoning the patients? Like, chemically?
That's what people feared at first, but subsequent deep toxicological analyses suggested it wasn't the fluorine metabolites themselves acting as a direct poison. Rather, the sheer burden of heavily modified, highly unnatural nucleic acids accumulating inside the cells eventually caused hepatotoxicity.
The liver cells essentially choked on all the synthetic material.
Exactly. It forced the entire industry to realize that maximum stability does not equal maximum safety.
They had to find a compromise.
And the compromise they engineered is called a checkerboard pattern.
A checkerboard pattern. Meaning what, exactly?
Instead of overloading the siRNA with one specific modification, chemists meticulously alternated 2-OMe and 2F nucleotides along the length of both the guide and passenger strands.
So it's like a methyl, then a fluoro, then a methyl, then a fluoro.
Yes, and by staggering them in this checkerboard, they hit the absolute sweet spot. They maintained the profound nucleus resistance and the immune cloaking of the methyl groups,
While also harnessing the enhanced binding affinity of the fluoro groups.
all while keeping the overall synthetic burden low enough that the cell could tolerate it long term.
Okay, so we've customized the sugar rings, but they also went after the actual spine of the molecule, right? The phosphate backbone that links the letters together.
Right. In a native RNA molecule, the nucleotides are connected by phosphodiester bonds. These are the specific bonds the nucleus wants to sever. To toughen this spine, chemists developed what are called phosphorothioate linkages, or PS bonds.
This is one of the coolest atomic tricks in the sources. A standard phosphate group has oxygen atoms sticking off it. The chemists took one of those non-bridging oxygen atoms and simply swapped it out for a sulfur atom.
It seems like a minor elemental swap, but the physical consequences are massive. Sulfur has a larger atomic radius than oxygen, and it changes the charge distribution of the bond.
Making it stronger.
Much stronger. A phosphorothioate linkage is roughly 100-fold more resistant to nucleus hydrolysis than a standard bond. It acts as an incredibly tough, impenetrable shield.
Where do they place these sulfur shields? On every single link?
No, they are most critical at the overhanging ends of the siRNA strands. Remember that dicer leaves a 2-nucleotide overhang at the 3' ends.
Right, those loose dangling ends.
Those loose ends are highly exposed, and they are the primary targets for exonucleases enzymes that chew away at RNA from the outside edges inward. By replacing the bonds at those exact ends with PS linkages, you seal off the most vulnerable attack vectors.
I always visualize this as replacing the flimsy plastic hinges on a door with solid titanium. It still swings open exactly the same way the RNA can still physically load into the AGO2 protein and function, but you absolutely cannot break it down with a hammer.
The titanium analogy is very apt, and there's an additional benefit to the sulfur. Because sulfur is less hydrophilic than oxygen, substituting it increases the hydrophobicity of the RNA molecule slightly.
Meaning it repels water a bit more, making it a bit more fat-like.
Exactly. And this encourages the siRNA to bind loosely to carrier proteins circulating in the blood, like albumin.
Wait, why is binding to blood proteins helpful?
Because a tiny 21-nucleotide piece of RNA is actually small enough to be rapidly filtered out of the blood by the kidneys and excreted in the urine. By hitching a ride on massive albumin proteins, the siRNA avoids renal clearance, significantly prolonging its circulation time in the body.
But just like with the fluorine, there's a catch to the titanium hinges, right? If you make the whole spine out of sulfur, it stops working.
You're right. Too many PS linkages stiffen the molecule too much. It severely reduces its binding affinity for the AGO2 protein. And if it can't load into RISC, it's completely useless.
Plus, heavy PS modification contributes to liver toxicity.
It does. So the standard ESC enhanced stabilization chemistry design limits these sulfur bonds to just 2 or 3 at the very terminal ends of the strands. It leaves the central core of the molecule native, so a go-to can perform its slicing action unhindered.
So clever. Are there any other advanced chemical tweaks they developed to push the limits even further?
The field is relentless in its optimization. Two advanced modifications frequently used are locked nucleic acids, or LNAs, and the 5-VP modification.
An LNA involves introducing a synthetic methylene bridge connecting the 2-oxygen to the 4-port carbon of the ribose ring, right?
Yes. It literally locks the ring structure in place.
It locks it into the ideal geometry for binding.
The thermal stability it provides is extreme, but because it binds so fiercely, it has to be used incredibly sparingly, often just one single nucleotide in the sequence. Otherwise, the siRNA becomes completely rigid or starts binding to off-target sequences.
Okay, and the 5-VP, what does that do?
That stands for 5-E vinyl phosphonate. If you recall, the dicer enzyme naturally leaves a phosphate group at the 5' end of the guide strand.
Right.
That specific phosphate acts as a key. It drops into a very specific binding pocket on the AGO2 protein to anchor the strand in place.
So if that phosphate falls off, the weapon won't load properly.
And unfortunately, our cells contain enzymes called phosphatases whose only job is to rip phosphate groups off molecules.
Of course they do.
The 5-VP modification is a brilliant piece of synthetic mimicry. It looks physically identical to a natural phosphate group, allowing it to anchor perfectly into a go-to, but its chemical bonds are completely immune to phosphatase enzymes.
It guarantees the guide strand remains permanently armed.
Exactly.
It is genuinely astounding. When you synthesize all these modifications together, the alternating checkerboard of methyl and fluoro sugars to balance stealth and potency, the titanium-sulfur bonds protecting the ends from being chewed up, the locked nucleic acids for grip, the vinyl phosphonate anchor ensuring it loads into the protein. I mean, we went from a naked biological molecule that dissolved in human blood in 5 minutes to an indestructible, programmable robot that can remain stable inside human tissue for months.
The chemistry is a total triumph. But here is the paradox. We built the perfect, indestructible therapeutic payload. But we've essentially built a tank that doesn't have the keys to get inside the enemy base.
Right, because of the cell membrane. Even with all the modifications, the siRNA is still a relatively massive molecule. It's about 14,000 Daltons. It has a highly negatively charged backbone from all those phosphates, and it is largely hydrophilic. It loves water.
And what surrounds every single cell in the human body?
A lipid bilayer, a dense membrane of fat that is heavily hydrophobic and uncharged.
The fundamental laws of biophysics dictate that a large, negatively charged, water-loving molecule physically cannot pass through a wall of fat. It will just bounce off.
Which brings us to the monumental challenge of delivery. The chemists built the payload, but the bioengineers had to build the transport vehicle to escort it across that cellular border.
Yes.
And the first true victory in that arena was the lipid nanoparticle, or LNP.
LNPs are famous now because of the mRNA vaccines, but the entire LNP architecture was actually pioneered and optimized over a decade specifically to solve the siRNA delivery problem.
Really? I feel like everyone thinks LNPs were invented for COVID.
Not at all. And an LNP is not just a microscopic drop of fat. It is a highly complex, multi-component delivery machine, typically built from 4 very specific lipid types.
Okay, let's break down the anatomy of this machine.
First, you have cholesterol. Just like in our own cell membranes, cholesterol provides essential structural rigidity to the nanoparticle, keeping it intact as it gets battered by the sheer forces of the bloodstream.
It's scaffolding.
Second, you have a helper lipid, often DSPC. Its geometric shape mimics the natural lipids of the cell membrane, which aids in the eventual fusion process.
Third is the PEG lipid, which is all about stealth, right? Polyethylene glycol?
Yes. The PEG lipid forms a hydrating protective shell around the exterior of the particle. This shell prevents multiple LNPs from clumping together in the vial, and crucially, it cloaks the particle from immune cells called macrophages that would otherwise swallow and destroy it in the blood.
So PEG ensures the LNP circulates long enough to find its target.
But the real magic, the actual engine that makes the whole system work, is the fourth component, the ionizable lipid.
The ionizable lipid. This is the key to crossing the membrane, isn't it?
It is. Ionizable means its electrical charge is dynamic. It changes depending on the pH of its surrounding environment.
Okay, so when the LNP is injected and circulating in the bloodstream, which has a relatively neutral pH of about 7.4, what is the charge?
The ionizable lipid remains completely uncharged. It's neutral.
Wait, why is that important? Why not just make it positively charged so it immediately sticks to the negatively charged cell membrane?
Because highly positively charged or cationic lipids are incredibly toxic to humans. They stick to everything in the blood, they rip apart red blood cells, and they are rapidly cleared by the liver and spleen. The ionizable lipid allows the LNP to cruise through the blood safely and stealthily.
So it's cruising through the blood, completely neutral. Eventually, it bumps into a target cell, let's say a hepatocyte in the liver. The liver cell's natural receptors grab the LNP and pull it inside through a process called endocytosis.
Right. The cell swallows the particle into a microscopic internal pocket called an endosome.
And this is where the vehicle has to perform its primary function. Because the endosome is essentially a cellular stomach. The cell's goal is to digest whatever it just swallowed. I really want to focus on the physics of this moment, because we can't just say the LNP escapes. If it just sits in the endosome, the cell is going to pump it full of acid and destroy our perfect molecular robot. How does a tiny sphere of fat actually shatter a cellular stomach?
It's a violent physical disruption. As the cell prepares to digest the LNP, it actively pumps protons into the endosome.
And protons carry a positive charge.
As protons flood in, the pH inside the endosome drops precipitously. It becomes highly acidic.
And that drop in pH is the trigger for our ionizable lipid.
Yes, the ionizable lipids are engineered with a specific pKa. As the environment becomes acidic, they act like sponges, absorbing those incoming protons.
Okay.
As they absorb protons, they undergo a rapid electrical shift. They transition from being completely neutral to highly aggressively positively charged.
And the interior wall of that endosome is lined with the cell's natural negatively charged lipids.
So now, confined inside this microscopic shrinking bubble, you suddenly have a massive buildup of positive LNP lipids pressing directly against the negative endosomal lipids.
Opposite charges attract.
They do, but in this confined space, they bind together violently.
It's a massive electrostatic collision.
The physical interaction between the positive ionizable lipids and the negative membrane lipids causes what we call a non-bilayer phase change.
Meaning what?
It completely ruins the parallel structure geometry of the endosomal membrane. The lipids contort into bulky, inverted micelle structures. The membrane physically shatters.
Wow.
The endosome tears open, and the siRNA payload spills out freely into the cytoplasm, exactly where the RNA machinery is waiting to load it.
It is literally a Trojan horse that explodes from the inside using the cell's own digestive acid as the detonator. It's brilliant.
It really is.
LNPs proved that RNA interference could actually work in humans. But looking at the source material, the pharmaceutical industry clearly wasn't satisfied with just LNPs.
Yeah.
They viewed them as a bulky, difficult first-generation technology.
Yeah, LNPs are effective, but they come with significant clinical baggage. They are complex and expensive to manufacture. They must be administered via a slow intravenous infusion in a clinical setting.
And despite the PEG cloaking, the lipid components can still trigger infusion-related inflammatory reactions in some patients, right? Like, they often require pre-medication with steroids.
Yes. The industry wanted to move away from these massive synthetic vehicles entirely. They wanted an address label approach.
Enter the GalNAc revolution, N-acetylgalactosamine. This feels like the biggest inflection point in the entire field.
It absolutely is. GalNAc is a highly specific sugar molecule, and its power derives entirely from its relationship with the receptor called the acetylglycoprotein receptor, or ASGPR.
That is a dense piece of jargon. ASGPR. What makes that receptor so special?
What makes it special is its location and its capacity. ASGPR is expressed almost exclusively on the surface of liver cells, or hepatocytes, and it is expressed in staggering numbers.
How many we're talking?
A single hepatocyte can have up to 1 million of these receptors coating its surface. Their natural biological function is to grab aging, damaged proteins out of the blood and pull them into the liver for destruction.
And what do these millions of receptors bind to?
They bind with incredibly high affinity to GalNAc sugars.
So the chemists realized, if we just take our naked, chemically indestructible siRNA and glue this specific sugar to the end of it, the liver will just absorb it automatically.
That was the exact hypothesis. But they quickly found that attaching a single GalNAc sugar molecule didn't provide quite enough gripping force. So they engineered a trivalent cluster.
3 GalNAc sugars arranged on a tiny branching molecular stem.
Right. When you attach this trivalent GalNAc cluster to an siRNA, it acts like subnanomolar molecular Velcro. It binds to the ASGPR receptor instantly and irreversibly.
And because the delivery vehicle is literally just a tiny cluster of 3 sugars, you don't need lipids anymore. You don't need cholesterol. The whole molecule remains incredibly small.
And this raises an important question about the clinical implications of that size reduction, which are massive. Because there are no lipids to cause an immune reaction, the patient doesn't need a massive 5e infusion.
So how do they take it?
You can dissolve the GalNAc siRNA in a tiny amount of saline and inject it subcutaneously, just under the skin, exactly like a diabetic using an insulin pen.
The patient can just do it themselves at home. It goes into the subcutaneous tissue, absorbs into the bloodstream, circulates to the liver, and immediately sticks to the ASGPR receptors.
And here is the truly elegant part of the system, the receptor kinetics.
Okay, what happened?
Once the ASGPR receptor binds the GalNAc drug and pulls it into the endosome, the receptor doesn't get destroyed. It detaches from the drug and recycles back to the surface of the cell in less than 15 minutes, ready to grab another molecule.
So it functions as a continuous, high-speed cellular conveyor belt.
Exactly, pulling massive quantities of the drug out of the blood and into the liver over several days.
Okay, wait, I have to make a deduction here. If GalNAc is just a sugar, and the drug doesn't have those ionizable lipids that change charge and burst the endosome, how does the siRNA actually get out of the endosome into the cytoplasm? Does it just get trapped?
You've zeroed in on the exact biophysical bottleneck of the GalNAc platform. The truth is, GalNAc delivery is catastrophically terrible at endosomal escape.
Really?
Yes. Without those disruptive lipids, there is no physical phase change to tear the membrane. The vast majority of the GalNAc siRNA gets permanently trapped inside the endosome and is eventually degraded by the cell.
How terrible are we talking? What percentage actually makes it out to find the RISC complex?
Quantitative studies show that less than 1% of the internalized GalNAc siRNA actually escapes the endosome into the cytoplasm.
Less than 1%. As an engineer, an efficiency rate of less than 1% sounds like a total system failure.
Yeah.
How is this the gold standard for therapy if 99% of the drug goes straight into the cellular trash can?
Because of the raw power of the chemistry we discussed earlier. The ASGPR conveyor belt pulls so much drug into the cell, and the ESC-modified siRNA is so chemically indestructible, and the ALGO2 protein is so catalytically efficient at slicing mRNA.
That the tiny 1% that does leak out is more than enough to completely saturate the RISC complexes.
Exactly. That tiny 1% can silence the target gene for up to 6 months.
Wow. They traded delivery efficiency for massive uptake capacity, extreme stability, and ultimate safety.
It's a fascinating compromise.
It really is. Okay, let's look at the actual results of that compromise. The chemistry is perfected. The delivery vehicles, LNPs, and GalNAc are operational. What happens when this hits the clinic? What diseases are we actually curing right now?
Well, the clinical validation of this entire field arrived in August 2018 with the FDA approval of patisiran, marketed as Onpetro.
And this was the first RNAi therapeutic ever approved, right?
Yes, and it targeted a truly devastating disease, hereditary transthyretin-mediated amyloidosis, or HATTR amyloidosis, with polyneuropathy.
Let's delve into the pathology of ATTR. What is actually happening to these patients?
It is a progressive, fatal genetic disorder. Patients possess a mutation in the TTR gene, which is primarily active in the liver.
And what does that gene normally do?
Its normal job is to produce a transport protein called transthyretin. But because of the mutation, the protein that gets manufactured is structurally unstable. It misfolds.
And misfolded proteins are very dangerous.
Extremely. These misfolded TTR proteins clump together, forming rigid, toxic amyloid fibrils. These microscopic shards of protein circulate in the blood and begin depositing in tissues all over the body, but they preferentially crush and destroy the peripheral nerves and the heart muscle.
So the patient experiences severe, agonizing, burning nerve pain. They lose the ability to walk. Their autonomic nervous system crashes so they can't digest food.
And eventually, the amyloid physically stiffens the heart until it fails, and traditional medicine had nothing for them.
The only viable treatment for decades was the full liver transplant, right? Literally cutting out the patient's liver and giving them a donor liver just to swap out the broken genetic factory.
It was highly risky and didn't even fix the amyloid that was already deposited in the nerves. But patisiran changed everything.
And patisiran uses the first-generation LNP delivery system.
Yes. The patient receives an intravenous infusion. The LNPs circulate, enter the liver cells, the ionizable lipids shatter the endosomes, and the siRNA payload is released into the cytoplasm.
And that specific siRNA is perfectly coded to match the mutant TTR messenger RNA.
Exactly. The RISC complex loads the guide strand, hunts down every single TTR message, and slices them to pieces before the misfolded protein can ever be built.
It just turns off the toxic factory at the source.
And the clinical trial data was miraculous. Not only did patisiran completely halt the progression of the devastating neuropathy, but because the constant flood of toxic protein was stopped, the patients' bodies were able to slowly clear the existing amyloid deposits.
Wait, really? The symptoms actually reversed?
For many patients, yes. They regained function. It proved that systemic RNAi was not just scientifically possible, but clinically transformative.
So patisiran proved the LNP model in 2018, but the industry was already pivoting to the sugar label approach. What was the first GalNAc drug across the finish line?
Just 1 year later, in 2019, the FDA approved givosiran, or Givlari. This was the validation of the entire GalNAc platform, and it targeted a disease that sounds like something out of a medical horror story: acute hepatic porphyria, or AHP.
Porphyria.
Yeah.
You know, there's historical speculation that the symptoms of this disease might have actually contributed to early vampire myths: severe sun sensitivity, receding gums, reddish-purple urine.
Yeah, but the reality for the patient is purely agonizing. It is characterized by sudden, utterly debilitating neurological attacks.
Oh, what a cause is it?
AHP is caused by a genetic defect in the biochemical pathway that the liver uses to synthesize heme, the iron-binding molecule essential for red blood cells and liver enzymes.
Okay, so it's like an assembly line. Step 1, step 2, step 3.
Exactly. In AHP patients, one of the enzymes further down the assembly line is defective or missing. Because the line is broken, massive traffic jams occur.
And those traffic jams create toxic intermediate chemicals.
Right. Specifically precursors called ALA and KG. They accumulate to massive toxic levels in the liver and spill into the blood. These precursors are highly neurotoxic. They attack the nervous system, causing abdominal pain so severe it often mimics an acute surgical emergency, along with paralysis, seizures, and intense psychiatric hallucinations.
But wait, if the disease is caused by a missing or defective enzyme, how does siRNA fix it? You can't use a genetic mute button to replace something that isn't there.
And this is where the biological logic of the drug design is just so brilliant. You don't try to replace the missing enzyme, you look upstream in the assembly line. Givosiran targets the ALAS1 gene, which is the very first enzyme in the heme synthesis pathway. It controls the rate of the entire factory.
So by muting LAS1, you intentionally slow down the beginning of the assembly line so the broken machine further down isn't overwhelmed. You stop the traffic jam before it starts.
Exactly. You restrict the flow, preventing those toxic ALA and PBG precursors from ever building up.
Yeah.
And because givosiran is a GalNAc conjugate, the delivery is trivial.
Right. Patients who used to spend weeks hospitalized on morphine for the pain attacks can now just give themselves a quick subcutaneous injection once a month.
The clinical trials showed a massive, near 90% reduction in those debilitating attacks. It gave them their lives back.
It's incredible. You just find the bottleneck and you shut off the valve feeding it.
Right.
The sources also mention lumasiran and nitazoxanide for a disease called primary hyperoxaluria type 1.
PH1 is another tragic assembly line defect, often presenting in very young children. The liver has a defect that causes it to vastly overproduce a substance called oxalate.
And the human body has no way to break down oxalate, right? So it all gets dumped into the blood and filtered by the kidneys.
And oxalate forms crystals. It forms massive, jagged calcium oxalate kidney stones continuously.
That sounds incredibly painful.
It is, and these stones physically destroy the delicate filtration architecture of the kidneys, rapidly leading to end-stage renal failure. These children often require dual liver kidney transplants just to survive.
And the siRNA approach is similar to porphyria. Target an upstream enzyme.
Yes. Lumasiran targets the HAO1 gene, and nitazoxanide targets the LDHA gene. Both of these are upstream enzymes in the liver that feed into oxalate production.
So by delivering these GalNAc siRNAs to the liver via a simple injection, you shut down the production of oxalate at the source, completely saving the patient's native kidneys from destruction.
That's right.
So we have HETR amyloidosis, AHP, and PH1. These are brilliant, life-saving therapies that perfectly demonstrate the mechanics we've discussed. But they are all ultra-rare orphan diseases. Has this programmable robot broken out of the rare disease space? Has it been used to treat common diseases that affect millions of people?
That boundary was absolutely shattered with the approval of inclisiran, marketed as Lephio. It was approved in Europe in 2020 and the US in 2021.
And what does inclisiran target?
It targets hypercholesterolemia, chronically high LDL cholesterol, which is one of the leading global drivers of cardiovascular disease, heart attacks, and strokes.
I mean, everyone knows someone taking a statin pill every day for high cholesterol. How does inclisiran change that paradigm?
It fundamentally changes the maintenance of the disease. Inclisiran targets a gene called PCSK9 in the liver. To understand why, you have to look at how the liver clears bad cholesterol.
Okay.
Liver cells have LDL receptors on their surface that act like molecular vacuums, pulling bad cholesterol out of the blood.
More vacuums, less cholesterol in the blood.
Right, but the PCSK9 protein is a natural regulator that binds to these LDL vacuums and destroys them, preventing the liver from clearing too much cholesterol. Some people have overactive PCSK9, meaning their vacuums are constantly being destroyed, leading to dangerously high blood cholesterol.
But we already have drugs that target PCSK9, right? Like monoclonal antibodies.
We do, but antibodies require the patient to inject themselves every 2 to 4 weeks, constantly battling the fully formed PCSK9 proteins in the blood. Inclisiran goes to the source.
It stops it from being made.
It is an advanced ESC GalNAc siRNA that enters the liver and slices the PCSK9 messenger RNA. The liver stops making the destructive protein entirely. The number of LDL vacuums on the surface of the liver skyrockets, and they continuously scrub the blood.
And because the chemistry is so durable.
A patient receives just 2 doses a year.
2 doses a year.
A single subcutaneous shot every 6 months provides persistent, round-the-clock suppression, cutting their LDL cholesterol levels by 50%. It completely removes the burden of daily pills or weekly injections for a chronic cardiovascular condition.
2 shots a year to functionally cure high cholesterol. That is wild. And the source material highlights another major approval hitting right now in 2025. Fitusiran for hemophilia A and B.
Fitusiran is perhaps the most elegant example of biological rebalancing using RNA interference.
Let's talk about hemophilia. It's a severe bleeding disorder. The patient is missing crucial proteins in the coagulation cascade, specifically factor VIII or factor IX.
Right, and because these factors are missing, their blood cannot form a stable clot, leading to spontaneous crippling bleeds into their joints, muscles, and organs.
The traditional treatment is factor replacement therapy, right? Hooking the patient up to an IV multiple times a week to infuse the missing protein.
Yes, which is an incredibly heavy burden on the patient. Now, as we noted before, siRNA cannot replace a missing gene. It is a mute button. So how do you use a mute button to fix a system that is already missing a critical component?
Well, you silence the counter-regulatory system.
Exactly. The human coagulation system is not a simple on-off switch. It is a delicate balance of pro-clotting proteins, the gas pedal, and anti-clotting proteins, the brakes. In a hemophiliac, they are missing the gas pedal, so the balance tips completely toward bleeding.
So fitusiran targets the brakes.
It specifically targets the SERPINS1 gene in the liver, which produces a protein called antithrombin. Antithrombin is one of the body's primary natural anticoagulants, a major brake on the system.
Okay.
By using a GalNAc siRNA to silence the production of antithrombin, fitusiran effectively removes the brakes. It lowers the threshold required for blood to clot, rebalancing the equation so that even without the missing factor VIII or INX, the patient's blood can form a stable clot and prevent spontaneous bleeding.
It's a masterstroke of physiological engineering. And because it's a GalNAc platform, it's just a simple subcutaneous injection, sometimes dosed as infrequently as once every 2 months, replacing hundreds of 4 infusions a year.
It really is life-changing.
You know, I want to pause here and speak directly to you, the listener. Think about the gravity of what we're discussing. Imagine living with a chronic, life-threatening condition, a disease whose management dictates your daily routine, your physical comfort, and your constant anxiety.
Yeah.
And the treatment isn't a handful of pills that cause systemic side effects or hours spent tethered to an IV pole. The treatment is a single tiny injection under the skin every few months that invisibly edits your body's cellular machinery at the source, preventing the biological error from ever occurring. That is not a science fiction premise. That is the reality of medicine today.
It is the full realization of the promise of the genomic era. But, as with all immense technological leaps, we have to acknowledge the boundaries. The system is not perfect, and it is not universally applicable yet.
Which leads us to the critical considerations, the ongoing challenges, and the immediate frontier of RNA interference.
Right.
Okay, time to play devil's advocate. We have a programmable molecular robot. We can input any 21-letter sequence to target any gene in the human body. The chemistry makes it completely indestructible. Why aren't we curing everything? Why is there no siRNA drug for Alzheimer's or muscular dystrophy or solid tumor cancers? What is the catch holding us back?
The catch, overwhelmingly, is the physical barrier of delivery. We have absolutely conquered the liver. GalNAc is a masterpiece of targeted delivery, but the vast majority of human diseases do not originate in the liver.
We need to break out. Extrahepatic delivery.
Precisely. The liver is the easiest organ to target because it naturally filters the entire blood volume and possesses millions of those recycling ASGPR receptors that we can easily hijack. Delivering a highly charged RNA molecule safely across the blood-brain barrier to reach a degenerating neuron, or pushing it deep into the chaotic, high-pressure microenvironment of a pancreatic tumor, or dispersing it evenly across the massive surface area of skeletal muscle tissue, that remains a staggering biophysical challenge.
Let's revisit that endosomal escape problem we talked about with GalNAc. We noted that in the liver, less than 1% of the drug actually escapes the endosome. The liver compensates because we can flood it with millions of molecules using the ASGPR conveyor belt. But if we are trying to target a muscle cell or a neuron, we won't have millions of specialized receptors to abuse. That 1% escape efficiency is going to be a hard stop, isn't it?
You've accurately diagnosed the primary rate-limiting step in extrahepatic delivery. We cannot rely on sheer volume outside the liver. Improving endosomal escape from 1% to even 10% or 20% would be biologically transformative.
So how are the bioengineers trying to force that endosome open without relying on massive, toxic lipid nanoparticles?
The current focus detailed in the sources is the use of cell-penetrating peptides, or CPPs.
Peptides.
These are short, specialized sequences of amino acids that possess the unique innate ability to physically push through lipid bilayer membranes.
So instead of a sugar label like GalNAc, you glue one of these peptide sequences to the siRNA.
Or you use them in tandem. You attach a targeting ligand to find the right cell, and you attach a CPP to handle the breakout. When the complex gets swallowed into the endosome, the CPP actively burrows into the endosomal membrane, creating pores or physically destabilizing the structure enough to let the siRNA payload slip through into the cytoplasm.
Oh, so it's a much more active escape mechanism than relying on passive leakage.
Exactly.
Okay, so we're working on cracking the endosome. What about safety and precision? We have this incredibly lethal a go-to slicer protein. What happens if our sniper rifle accidentally shoots the wrong target?
Off-target effects are a profound toxicological concern, and they trace back directly to our discussion comparing siRNA to microRNA.
Right, the sniper versus the broad network regulator.
Exactly. Remember that a microRNA only needs a tiny partial match, a 7-nucleotide sequence called the seed region, to bind to a messenger RNA and suppress it. If the synthetic siRNA we engineer happens to have a seed region that accidentally matches the sequence of an unintended, totally unrelated gene, the siRNA can inadvertently act like a microRNA.
It might not slice the unintended gene, but it will sit on it and block it from being translated.
Yes, and if it does that to dozens of critical cellular pathways simultaneously, it causes massive transcriptional dysregulation.
The cell loses control of its own biology.
Right, which is a primary driver of the severe liver toxicity observed in early preclinical trials.
So how do the chemists prevent the siRNA from acting like a shotgun? How do they force it to only accept a 100% perfect match before binding?
The solution is purely chemical. Chemists return to the structure of that specific 2 to 8 nucleotide seed region on the guide strand. They intentionally introduce thermal destabilizing modifications right into that critical zone.
Thermal destabilization, meaning they make the bonds physically weaker or more awkward.
Exactly. They might add a bulky chemical group or utilize a specific glycol nucleic acid modification that distorts the geometry just enough that partial binding becomes thermodynamically impossible.
Oh, that's smart.
The physical barrier is so high that the siRNA will only bind if the entire 21 nucleotide sequence lines up perfectly to overcome the resistance.
So if it's not a perfect match, it just won't stick at all.
If it's not a 100% perfect match, the modified seed region refuses to stick. It eliminates the microRNA-like off-target toxicity entirely.
It's astounding how every biological problem ultimately has a structural chemical solution. Are there any actual breakthroughs showing we can get into these tough extrahepatic tissues?
We are seeing significant early victories. To cross the blood-brain barrier and penetrate the central nervous system, researchers have developed C16 lipid conjugates.
C16, meaning a carbon chain.
Yes, a specific, highly lipophilic 16-carbon fatty acid attached directly to the siRNA. This fatty chain allows the molecule to interact favorably with the dense lipid environment of neural tissue, facilitating deep tissue penetration and cellular uptake in the brain and spinal cord.
Which opens the door for targeting neurodegenerative diseases.
Absolutely.
And we absolutely cannot have a discussion about the frontier of genetic medicine without talking about oncology. Is this programmable assassin being deployed against cancer?
Cancer is the ultimate target, but solid tumors present a nightmare delivery scenario. They are dense, fibrotic, chaotic masses of tissue with terrible, irregular blood supply.
So if you inject siRNAs systemically into the vein, it is incredibly difficult to achieve therapeutic concentrations deep inside the tumor microenvironment. The blood simply doesn't flow there effectively.
Right, but the source material outlines a fascinating clinical trial that bypasses the bloodstream entirely.
Tell me about SERL-204.
The sources highlight this clinical product, SIL-204, which takes a radically different physical approach. It is an extended-release siRNA polymeric implant specifically designed for patients with locally advanced pancreatic cancer.
Pancreatic cancer is notoriously aggressive and highly resistant to traditional chemotherapy.
It is a devastating diagnosis, but instead of trying to circulate the drug, the surgeons physically implant this miniaturized matrix directly into the core of the pancreatic tumor.
Oh, directly into the tumor itself.
Yes. The implant then slowly, continuously elutes the siRNA directly into the surrounding cancer cells over the course of months.
And what specific gene is the siRNA targeting inside those cancer cells?
It is specifically coded to silence the KRAS gene.
KRAS.
Yeah.
That is one of the most famous oncogenes in all of biology. It's a major driver of tumor growth.
Yes. Specifically, this siRNA targets the mutant G12D and G12V variants of KRAS. In pancreatic cancer, these mutated genes act like a gas pedal permanently glued to the floor. They're constantly screaming at the cell to divide, grow, and invade.
So the implant releases the siRNA, it loads into the RISC complex inside the cancer cells, and it slices the KRAS messenger RNA to pieces.
It completely severs the fuel line to the tumor. It mutes the signal driving the uncontrolled division.
And because the delivery is highly localized, systemic toxicity is minimized.
Right, and the directional trends in tumor response and patient survival in these early trials are incredibly promising. It proves that if we can solve the spatial delivery problem, the siRNA payload itself is more than capable of executing the target.
If we pull back and synthesize everything we've explored today, the trajectory of this technology is staggering.
It is one of the most rapid, profound arcs in modern science. Just over 20 years ago, siRNA was nothing more than a basic research tool for functional genomics. Scientists were using massive libraries of synthetic siRNA in petri dishes to systematically knock down genes one by one, just to observe what happened and figure out what those genes did.
Then it evolved into a highly complex intravenous LNP therapy for an ultra-rare orphan disease like hATTR amyloidosis.
Then, with the brilliant simplicity of the GalNAc sugar label, it broke into the mainstream, offering twice-a-year treatments for common global conditions like hypercholesterolemia.
And now, armed with cell-penetrating peptides, C16 lipids, and localized tumor implants, we were standing on the frontier of targeting the central nervous system, muscle tissue, and the fundamental genetic drivers of oncology.
It's an incredible time for medicine.
We have covered an immense amount of scientific ground today. We began with botanists utterly confused by the appearance of white petunias. We moved to microscopic worms that revealed a hidden, ancient genetic immune system designed to shred viral RNA.
We watched as medicinal chemists took a fragile biological signal that dissolved in human blood in 5 minutes and meticulously armored it.
Armored it with methyl stealth cloaks and titanium sulfur bonds, engineering a truly indestructible molecular robot. We unpacked the biophysics of endosomal escape, moving from the violent electrostatic explosions of lipid nanoparticles to the elegant high-capacity receptor conveyor belts of GalNAc.
And we have seen how this sequence of 21 letters is quite literally rewriting the biological fate of patients suffering from diseases that were previously deemed untreatable.
The clinical reality of drugs like patisiran, givosiran, lumasiran, and inclisiran absolutely cements the fact that genetic medicine is no longer a speculative future promise.
It is an established, validated, and highly potent therapeutic modality operating in clinics worldwide today.
My hope is that this deep dive has served as a powerful conceptual shortcut for you. You are now equipped with the mechanical understanding of the very cutting edge of precision medicine. Keep a close eye on this space. Watch for those breakthroughs in extrahepatic delivery platforms, the peptides and the lipid conjugates, because as this technology matures, it is inevitably going to touch your healthcare or the healthcare of someone you love in the near future.
And as we wrap up this exploration, I want to leave you with one final provocative concept to mull over. We have spent the last hour discussing how we can perfectly program a drug to selectively silence disease-causing mutations with unparalleled precision.
Right, fixing broken assembly lines and misfolded proteins?
But consider the broader implications. If we now possess the validated technology to program a durable, safe molecular machine that can selectively silence any active gene in the human body simply by inputting a specific 21-letter sequence, what happens when we shift our focus?
Shift our focus to what?
What happens when we move beyond targeting rare genetic errors and begin identifying and targeting the specific genes that dictate the fundamental processes of cellular senescence and aging itself? If biological aging is, at least in part, driven by a specific cascade of genetic expressions over time, Sarah might just hold the biological mute button.
Yeah, there's a thought to keep you up at night. Until next time.